Hello everyone, welcome to my channel Anytime Medicine, and we will going to talk about purine metabolism in these slides. So what are the basic nucleotide roles in human body? They are RNA and DNA monomers. They are source of energy like ATP. They work as physiological mediator like cyclic AMP and cyclic GMP. Cyclic AMP is a blood flow regulator and cyclic GMP is a secondary messenger in signaling pathway. What are the source of nucleotides? We can get nucleotides by two ways, exogenously and endogenously. Exogenously we can get it by diet, and endogenously we can get it by biochemical synthesis. We can get it by salvage or direct synthesis. We are going to talk about each in these slides. Remember, RNA synthesized first and then RNA converted into the DNA. There are two different pathways for purine and pyrimidine synthesis. In nucleotide, all nitrogens are come from amino acids. So what are the purines? Purine has two rings. There are two purine structures, adenine and guanine. Here you can see the structures of adenine and guanine. Purine synthesis. What we need for purine synthesis? Mainly three things: ribose phosphate, amino acid and carbons. Ribose phosphate, which comes from HMP shunt, carbon, which is come from tetrahydrofolate, and carbon dioxides. Our goal is to create IMP, inosine monophosphate. In order to create IMP, we have to first create PRPP, that stands for 5-phosphoribosal 1-pyrophosphate. Here you can see the reaction. Ribose 5-phosphate is converting into the PRPP. Here you can see the structure of PRPP. PRPP has two phosphate groups in it, and that two phosphate group comes from ATP. And here you can see the ATP is being used in these reactions. So now you can see two phosphate group in PRPP. In our previous slide, we have seen how PRPP is created. Now PRPP will go through some series of reactions and will create IMP. Here you can see the structure of PRPP and IMP. In a structure of IMP, you can see the xanthine structure that we will talk about in next slides. IMP structure is very much like nucleotide, and therefore it is called an intermediate nucleotide. IMP has amino group, phosphate group and a base like any other nucleotide. In this structure, base is hypoxanthine. Note that in hypoxanthine, carbons and nitrogens are come from amino acid, folate and carbon dioxide. Purine has two rings. One is 5-unit ring which has two double bonds, and other has 6-unit rings and which has three double bonds. Remember, in purine rings, nitrogen sources are aspartate, glycine, and glutamate, and the carbon sources are carbon dioxide, glycine, and tetrahydrofolate. There is contribution of folate too in purine synthesis. So, how the regulation of synthesis occur? We have already seen that ribose 5-phosphate will convert into the PRPP, and then PRPP will go through some series of reaction and will convert into the IMP. In order to create IMP, glutamine PRPP aminotransferase will help, and IMP will convert into the AMP and GMP. So now IMP and GMP and AMP will itself control the or regulate the synthesis of purine synthesis. Here you can see the high amount of IMP, GMP and AMP will give the negative feedback to the enzyme, and that will slow down the reactions. Now we will see the rate limiting step of purine synthesis. Now IMP converts into the XMP xanthinosin monophosphate via enzyme called IMP dehydrogenase or IMPDH with the help of NAD plus and H2O. Here you can see the reaction of IMP converting into the XMP with the enzyme called IMPDH. There are certain IMPDH inhibitors that will stop the reaction, and we will see that in my next video. Now the XMP converts into the GMP with the help of glutamine. So IMPDH is a rate limiting step for GMP synthesis, therefore regulatory step for DNA synthesis. So in purine synthesis, now IMP and GMP will convert into the ADP and GDP respectively by adding the 1-phosphate into their structures. ADP and GDP stands for adenosine diphosphate and guanosine diphosphate. Then ADP and GDP will convert into their deoxy form, deoxy-ADP and deoxy-GDP respectively via enzyme called ribonucleotide reductase. Here you can see the reaction. ADP is converting into the ADP and GDP is converting into the GADP with the enzyme ribonucleotide reductase. We have talked about the purine synthesis, and now we will talk about the purine salvage, how the purine salvage works. Free floating amino acid will be salvaged or converted back into the nucleotide, or they will become uric acid and will be excreted from the body. We will going to first talk about hypoxanthine and guanine. Hypoxanthine and guanine will convert back into the IMP and GMP respectively, interacting with the PRPP and an enzyme called HGPRT. HGPRT stands for hypoxanthine guanine phosphoribotransferase. Hypoxanthine, here you can see the reaction hypoxanthine and guanine is interacting with the PRPP and convert into the IMP and GMP with the enzyme HGPRT. Adenine will have different pathways than guanine and hypoxanthine. Adenine will convert back into AMP interacting with the same PRPP and help of enzyme called adenosine phosphoribosyl transferase. Here you can see the reaction of it. Now how the purine breakdown occurs. Purine breakdowns into uric acid and will be excreted from the body. Hypoxanthine and guanine will convert into the xanthine with the help of enzyme called xanthine oxidase and guanine respectively. If the purine cannot break down into uric acid or uric acid cannot excrete from the body, that will have certain disorder that we will talk about in my next video. Now xanthine will become uric acid with the help of xanthine oxidase. If we have xanthine oxidase deficiency, we will have gout disease. That also we will talk about in my next video. In case of adenine, adenine will have to convert back into the nucleotide and then breakdown will occur. Adenine will first convert into the AMP and with enzyme called APRT, that stands for adenine phosphoribosyl transferase. Then AMP will convert into the adenosine. Adenosine will convert into the inosine with the help of adenosine deaminase. Inosine will convert into the hypoxanthine with the help of purine nucleotide phosphorylase. The hypoxanthine will undergo same reaction as we have seen in guanine and hypoxanthine breakdown and will eventually become uric acid. As I said earlier, adenine will go through series of reaction and the end product of that reaction will be hypoxanthine, and that hypoxanthine will undergo same reaction as we have talked about in the case of guanine and hypoxanthine. Thank you guys for watching my video. See you until next, and please do subscribe my channel.